Hello, I'm Dr. Leanne Taylor-Smith and I'm going to be showing you how to use the microscopes we have here in the teaching labs. I'll start by showing you how to take the compound microscopes out of the cupboards that they're kept in the lab. The microscopes are kept in cupboards underneath the benches with the handles facing out. Pick up the microscope by the handle, pull it out until you can get your hand underneath the front base of the microscope. Then carefully bring it up and place it on the bench. Ideally, place it on the bench in front of a position where you can sit at it and use the microscope easily. You can then take the cover off. And now we want to turn it so it's in position for use. Make sure that you lift it whenever you need to move it, rather than dragging it, because this can vibrate and damage the optics inside the microscope. And then untwist the cable at the back and plug that in. The microscope is now ready to use. So I'll start by going through some of the basic parts of the microscope. So starting at the top, these are the eyepieces. These are the parts that you need to look through to see your specimen, and they're also adjustable. You want to be able to adjust these so that you can look down both of them and see one circular field of view. Okay? So adjust these until they suit you. Next we come down to what we call the nose piece, which is where all of the objectives are kept. On this particular microscope we have a 4x objective, we have a 10x objective, we have a 40x phase objective that I'll come back to and tell you some more detail about later, and also a 100x objective, and that requires oil immersion to use. Next we have the slide holder. When we need to put a slide in, we just pull back on the slide holder there, push the slide in and let it grip the slide. We then have the stage. Now the stage movement is controlled by an XY controller here on the side, to be able to move your specimen and slide. Underneath that we have the condenser, and in the condenser is a diaphragm, an iris diaphragm, and this can be opened or closed and has markers on it specifically where it should be for the objective in use. Below that is the lamp housing where the LED light source is kept. On the right hand side here we have the on-off switch. We also have the coarse and the fine focus. And on the left side we have the controller for the level of light. Before we switch on the microscope, we want to make sure that the light level on this left hand side is down to its lowest point, and then we want to switch on this right side. And now the microscope's ready to use. What I'm going to do now is show you how to make a basic wet mount slide. So first of all, we need to get some of the equipment into place and have it all in a place where it's nice and easy to work with. So we've got some sterile distilled water, an inoculation loop, our microbiological specimen, which is some yeast for today, lighter for the Bunsen burner, some cover slips, and some microscope slides. So first of all, I'm going to get a microscope slide out so it's ready to work on. Our cover slips are there ready. So the first thing that we need to do is light the Bunsen. And remember that while we're working in an aseptic manner, we need to have the Bunsen on blue flame. And then we need to work as close to it as possible. So what we need to do first is provide some media for our sample on the slide, and I'm just going to put a few drops of sterile distilled water on here. So first of all, I'm going to open my bijou with my little finger, flame the neck, flame my loop, touch the side of the bottle, go into the water, take a loop full of water, tap that on, and do this just a couple of times to get some water onto the slide. I can then flame the neck, do my bottle back up, and flow my loop. Okay, and now we want to take up a very small amount of our yeast. So to do this, we flame our loop, cool it on the side of the agar first to take the heat out, pick up a small amount of our sample, place that back into the lid, and then simply just smear some of that sample into the water that we've put on there. Flame the loop again so that it's uncontaminated and safe. And the last thing to do is just to pop a cover slip on top. Try not to touch the middle of the cover slip. We want no fingerprints on it, so hold the edge of the cover slip, put it down one edge first, and then drop the cover slip. And the water will spread and hold the cover slip on the slide. So now we need to load our specimen onto the microscope. So to do this, we just pull back the holder there, slide our slide in, and let go. This will then hold this in place and allow us to move our cover slip around. So center our specimen, and what we're going to do is start at the lowest magnification first and then work our way up. A good rule of thumb to find your sample quickly is to start with the stage up at its highest point, which it is here. You should be able to find your specimen within 1.5 turns from this top position. We'll then check that our aperture is at the right level under the stage. We're set to the 4 times aperture for our 4 times objective. We then look down the eyepieces. Our light levels are fine. We now move down in focus to find our specimen and nicely focus on the specimen that we want to look at. Once we're happy with 4 times, we can then move up to 10 times. These microscopes are very good actually, and you should require minimal, very small amounts of focus change. Once we're happy at 10 times, we can then also go up to 40 times. Now the 40 times has got phase ability with this pH that's on the objective. So for this, we need to make sure again that the aperture underneath is allowing enough light through, and we also need to push our phase ring in underneath here. So under the stage, we push this in and you'll hear it click into place. And again, we can find our sample. We may need to add more light for phase as we've gone up in magnification and introduced a phase ring, and then we can find our sample again. Now, phase contrast is a technique which allows us to see transparent cells. So most living cells are transparent and have no colour. The very good advantage of phase contrast is that we can see living cells. So we don't need to fix or kill our cells or stain them before we visualise them. And phase contrast is actually used in some of the research labs in the university, such as the May lab, where we can look at macrophages and pathogenic fungus, and we can do time-lapse imaging overnight on microscopes. And that's because phase contrast allows us to look at living cells for long periods of time. Now, phase contrast works by exaggerating differences in the light as it passes through different specimens. So as the light passes through a cell, it has a different refractive index as to when it passes through the water around that cell. And this is exaggerated and transformed into a contrast that we can see down the eyepieces. What we can also look at is something a lot smaller, and that's something like a bacterial sample. So for bacteria, we would want to go up to 100 times slide. So what we can do is look at, for instance, a gram stain. And this is an example of a slide in which the cells have had to be killed and stained first before we can see them. And with the gram stain, we can see whether a particular bacterial pathogen is of gram-positive or gram-negative characteristic. Now this particular slide, we will start again. We'll start it 4 times again, bring our stage back up to the top, bring our aperture back down, take our phase ring back out, and probably turn the light down a little bit more. And we'll find ourselves at 4 times first. Okay, and again we can work our way up through the magnification. So 10 times, small adjustment on the focus to find our sample again. We can go to 40 times, and we can add the phase as well and increase our light again if we'd like to see these in phase contrast. And again, a small adjustment in focus. And now, if we want to move to 100 times and use the oil objective, then we need to be really sure that we're completely happy with our sample and our focus at 40 times before we move to 100 times. The oil that we use for the 100 times cannot go onto a dry lens. It can dry on and even damage any dry objectives that we're using. So we want to be absolutely sure that we're ready to start using oil. Now, I'm now happy with the focus that I have at this stage. If I now want to use oil, we use these small bottles that are labelled immersion oil. Now, to do this, a very good technique to do is to turn the nose piece so that we're at the halfway position between 40 and 100. We then put a very small amount onto our specimen using the dropper. So where you can see the light coming through, a drop of oil. And because we've focused it very well at 40 times, we can now swing the 100 times in and it will be touching the oil. Again, we may need to open the aperture slightly more. We can also now take out the phase ring because 100 times lens is not phase lens. A slight adjustment, and now we can see our nicely stained gram-negative E. coli. So the really important part now that we're finished using an oil objective is to clean the oil off so that we don't accidentally get any oil on any of the dry objectives. Now we do this not using the normal tissue that's in the lab, but using lens cloth. And this is special fine tissue that's designed for cleaning objectives without damaging them. So we again can go to the halfway position. We can take our slide out, and now we can swing the 40 back in, allowing us to get to the 100 times. We can then wipe the oil from the 100 times lens, so that is now clean. And it's also a good idea to remove it from the specimen that you've been looking at, to keep this clean so that other people can use it. So now, before we finish, we thought we'd go through some common mistakes for people who have troubles using the microscopes or can't find their specimen. A typical issue is that the stage is very low. Now, if we go by and see students where the stage is very low and they're struggling to find their specimen, it's probably just because the stage is too low. So you need to raise the stage up, start from a very high position. The focal plane should be only 1.5 turns from the top position of the stage. Another common mistake is that the phase ring is in when it shouldn't be. For instance, on the lower objectives. So on the 4 times and the 10 times, this phase ring should be out. One other problem can be that the iris diaphragm underneath is turned down too low or too high, meaning that the light levels aren't correct for you to be able to see your specimen. And finally, one last problem can be that the slide isn't held in position correctly. Sometimes it can be wedged underneath, and sometimes it can be raised on one side because it's not being held in and it's actually on top of the holder rather than being held in by it. And these are just a few things that are some common mistakes that you can look out for when you're using the microscope. So, now that we've finished using the microscope, we want to make sure that the phase ring is out for the next user. We want to make sure that the light levels are down, and then we can switch it off. We can then wrap up the cord on the back, which goes nicely into the holder here at the back. And remember to put the cover back on. We then place the microscope back into the cupboards underneath the benches, again making sure that you hold the handle and the front base. And that's how we use the compound microscopes in the teaching labs.